Oh,
Yes, okay, I'm recording.
okay. Great. So thank you, Laureline, for joining us today. So Laureline Logiaco is currently, well, did her postdoc. I know your career from your postdoc moment when you were at Columbia with Sean Escola. And now you're at MIT as a research scientist, and you're about to start your faculty position at Colorado and Schultz within the next few months. And we're very happy to have you here today. You're going to tell us about this recent chapter about models of thalamocortical interactions in motor control, and maybe partially how it relates to sequence is clearly something that a lot of us are interested in as well. So thank you again for joining us, and we're going to turn it over to you.
Thank you very much. And it's a relatively broad chapter review. So I think that there will be many possible, perspectives on what I'm going to talk about. So please interrupt me at any time. To start, because because I'm going to talk about different types of models and think about what they can tell us about thalamocortical interactions. I thought it was important to mention that there are really different types of models that really have different characteristics. And one important characteristic that can define a model can be scale. I'm not really necessarily talking here about Morse description levels. And rather than being a little bit unclear about that when thinking about our own field, which is still in development, I'd like to take an example that's historical, where we actually know what happened. Where there were several different models that were interested in phenotype variations across generations. At a larger scale, all of us know about Darwinian evolution theory, with the idea that you're going to have some mechanism that's not very well defined at the fine level of variation across animals, for instance, mice of different colors. Then you're going to have selection. Say a bird is going to eat the lighter mice more often. And then after inheritance and reproduction, you get more black mice. So that's, a larger scale description. At a more intermediate scale, we know about this scientific inquiry into inherited factors by Mendel's, which is really getting into more detail of the inheritance, with the idea that there is both recessive and dominant factors. And that explains why, for instance, a recessive color factor can skip a generation and only found in a quarter of the descendants at the second generation. And of course, more recently, over the course of the 20th century, there has been a lot of investigation about molecular basis of a lot of these phenomena, from the molecular basis of factors, alleles of genes, molecular mechanisms of variations and transmissions. And really, in my opinion, we can think of all of these models as being useful in different ways, and having different scopes, even though they share a thematic. So this is one way that models can vary. Another way that they can vary is the building principle, from what I would call empirical to more mechanistic. So in an empirical type of model, you're going to fit many parameters to observations. For instance, you can optimize an artificial neural network these days to fit neural activity or behavior. And this is a recent example from the literature where what they fit is the behavior of a fly, and not just in baseline conditions, but also in response to specific perturbations of specific units in the fly that corresponded to different units of their fitted models. On the other hand, you could have a more mechanistic type of building principle where you build your model by assembling building blocks with hypotheses and study resulting properties. For instance, this idea is that you can build V1 neurons response from LGN inputs, and you can think about the change in receptive field as a superposition principle. And they both have different characteristics. Of course, by definition on the left, the quantitative fit is going to be great. Versus on the right, typically it's going to be very approximate. On the left, you may have knobs to predict causal manipulations if those knobs are already present in the data and if the manipulations are close to the training data. Whereas on the right, the knobs are here by construction because you built blocks to try to make your model. So for instance, here, you have a very clear idea of what to expect if you inhibit one of these LGN cells. And on the left, it's going to be hard to adjust a parsimony accuracy trade-off. With current machine learning methods, typically you need very overparameterized types of neural networks to get good results. Whereas that was on the left, whereas on the right for mechanistic models, you could decide that you're going to make a smaller number of hypotheses to build your models. And you know that it's going to come at a cost of not capturing as much of the data, but maybe you think that you've captured most of the important things. Like if you were starting to include more hypotheses, then you need many more of these for a relatively smaller gain. So that's a second axis. And a third axis would be the main modeling goal, from descriptive to functional. So a descriptive model, for instance, can be these LFADS methods, where here what you're trying to do is to have a model that's going to be able to very accurately match single trial firing rate, for instance, versus on the right, a very famous example is this work by Yemins and DiCarlo, where in general, the question you're trying to answer with this model is question of the type, are firing rates the way they are because of circuit function? So in the case of the visual hierarchies, the idea is that the circuit function is to extract useful statistics from natural images. And so you can implement this circuit function in an artificial neural network. And the question is then, is this functional requirement leading you to similar activity in your network? So why did I talk about all of this? It's because we are going to review several types of models, and I wanted to be a little bit clear about what types of model we were focusing on, and also we found just examples of. So we focused on thalamocortical models for motor control. And really, that already, by the way they're called, suggests that a mesoscale type of focus is a relevant one, where you're going to respect architectural constraint within and between regions, with this idea that cortex is going to be this large recurrent network versus thalamus is going to be smaller and won't have at least excitatory recurrence. So it's not very functionally recurrent. In terms of building principles, because we tried to find the models that were available to us so far, most of the models that actually had been developed were more mechanistic, and the modeling goals varied a lot. So within that scope, what Sean and Koshek, that wrote the chapter with me, did is to then propose further separations or access to understand the space of motor thalamocortical models. And specifically, we proposed two different main axes. On one axis, the horizontal one here, we are going to basically think of cortical activity as having more of a representational perspective. So you're going to represent, for instance, reach angle, reach speed. These are more like abstract movement parameters, and the idea is that there is this abstract high-level representation that's going to instruct downstream circuit. On the right, this is a more dynamical perspective where the activity in motor cortex is thought to self-generate time-dependent command that typically is thought to then go more directly to towards the spinal cord and the muscles. And the second axis, which maybe is a little more binary, but everything in the end can, you can think also of ways in which it can be a little more continuous. It's the question of whether the model thinks that there is a lot of importance for the feedback from cortex to thalamus. So here, thalamus is a feed-forward input to cortex, versus models that assume that it's really loops between cortex and thalamus. And so what we did in this chapter is that we reviewed these different types of models, and I'm going to really tackle them in a series from type 1, type 2, and type 3 models that are in 3 quadrants of this graph, starting with type 1 models. And so in type 1 models, we think of thalamus as a feed-forward processing stage to a cortical representation. And really, I'm going to talk about both observations that led to this type of models, which I think started in pretty early studies, which really analyzed data in a way that was, I think, inspired or at least similar to early V1 LGN studies. And then we're going to talk about the associated models. So in terms of observations, I don't need to tell you in details that there has been a whole zoo of studies that focused on recording motor cortical activity while monkeys reach to different targets of different angles. And the first ways that people characterized the activity of neurons in motor cortex, that at least I know of, is to look at the firing rate of a motor cortical neuron across different reach angles. What struck people, or the main characteristics that they focused on, was that the firing rate in one cortical neurons could be thought as having a maximal response at a preferred angle on average, and less response away from this angle. And people recorded similarly in thalamus, and things seem to be perhaps not as already developed into really a preferred angle in the way this was reported at the time. And so the idea here is that motor cortex may represent abstract instructions. And one caveat that new results and studies have pointed out is that this view on its own may not easily explain the fact that in motor cortex there is fast time dependent of firing rate, even faster than the latency of sensory feedback. And perhaps even more importantly, we're going to talk about that later, but there has been a characterization in motor cortex activity of specific proportion and trigger of neural signatures before a movement bout. And so these are phenomena that are not really understood with that static view. And so the associated models here, I'm showing models by Kenji Doya, are going to be typically separated by which brain region project to the thalamus. But I kind of hid all of this in terms of whether basal ganglia or cerebellum projects to thalamus, because when we're trying to understand thalamocortical interactions here, in both of these models, really what we're having is that thalamus is a relay. These models, as you can see, do not leverage fast local recurrence for cortical common generation. And so my understanding of the hypothesis here is that if you have learned movements, that implicitly assumes that motor cortex has this like higher level common, let's say, reach direction, and it transmits it to downstream recurrent subcortical circuits. And so the assumption is that these recurrent subcortical circuits are the ones that are going to be able to be plastic, high capacity, and learning interference resistant. So that's maybe something to think about, especially given that mammals evolve quasi-direct projection from cortex towards the spinal cord. And so, as I mentioned, also it's at odds with some observations of motor cortical activity. So these were more like type 1 models, and a different category of models is going to include some local recurrence in motor cortex, but thalamus remains a feed-forward input to this recurrent cortex. So thalamus as an afferent to dynamical cortical models. So as a general framework, this is how you can think about it. Cortex is now going to be this dynamical system. It is going to have neural activity that is going to be defined or described as a differential equation. So that describes how the activity H changes over time. An example of such a general idea of a time-dependent thalamic input to a recurrent cortex was in models of MD thalamus and frontal eye field, with the idea that thalamus would transmit saccade-related information, and the frontal eye field would integrate this saccade-related information with visual information to yield consistent visual signals. So that's one example. There has been further examples more on the premotor side, with the idea that the thalamic input was going to then act more of a nonlinear gate modulator. So you're going to have in specific contexts, specific thalamic units that are active, and that would gain up or down cortical populations, and that leads to context-specific neural trajectories. And you can also push that to the extreme. And in this work, really, thalamus was a complete nonlinear selector. So it basically says one population of neurons is either on or off. And what these models, however, are not necessarily addressing is that there is anatomical and in some cases optogenetics evidence for the importance of the feedback from cortex to thalamus. If you're really thinking of thalamus as sending a feed-forward input, this only offers a few parameters. If you think of synapses as parameters that can be learned to perform a function. And finally, if you really are interested in motor execution, these mechanisms of gating and selection are a little hard to reconcile with the fact that the response of neurons in motor cortex don't seem to be super selective to one specific type of movement. They seem to be more mixed. So these were type 2 models. And now we're going to go towards type 3 models. So
Well, we can hear. Can you hear us? Could be.
Sorry, you haven't been able to hear me the whole time?
No. It was just this new slide, type 3 model. Yeah, I
Oh,
think
okay.
for a sec we got disconnected.
Okay.
Yeah, just like from this type 3 model. We had you until type 2.
Until type 3 model. So good.
Yeah.
So let me come back here. Okay, so the idea here is that we're going to have in these type 3 models a recurrent cortical network, and it's going to interact with the thalamus through loops. And really what these models, the common principle of these models is that cortex is going to be sort of a baseline output generator, and thalamus is going to modulate it and reconfigure it. And so there has been two major recent modeling approaches with complementary strengths to try to make sense of this setup. And one of them relies technically on lowering dynamic perturbation, and the other one on optimal control theory. So I'm going to try to, at a high level, give an intuition of how they are different. So in the case of lowering dynamic perturbation, the idea is that we have a baseline recurrent cortical network, if you want, that is fixed. And then we're asking, if we have only a few thalamic loop, which effectively are going to act as small in terms of number of parameters changes of the effective connectivity, how does that change our dynamics? And so in this framework, really, we take the small thalamic size as an important constraint from the get-go. And what we're interested in is in clarifying the power and the limitations of using a few thalamocortical loops that we adjust in order to reshape the dynamics. And the conclusion from studying this problem is that if what you want your network to do is to produce a relatively low dimensional output, for instance, motor output is relatively low dimensional, you have many fewer muscles than you have neurons, then even a single loop can largely expand the output repertoire compared to what you could do with your baseline network. However, if you have more higher dimensional constraints, let's say you want to fix all of the activity in all of the units, then you're going to need more loops. And this is actually a non-standard mathematical framework. There are lots of people actually from pretty different fields that are working on this. And so this is still in development. And so on the right, what I mentioned was optimal control theory as an alternative framework that people have used to try to start making sense of this. I'm just showing here a very general explanation of what the framework is about. So the idea is that you have a dynamical system with state variables. So you can think of it as the activity of cortex X. And it is governed by recurrent interactions. You can think of it as your recurrent connectivity matrix. A, which is fixed, it receives an external input U through potentially constrained directions B that are also fixed. And then you can add some noise. And the idea is that optimal control gives us a way to know what the input U should be if you want to optimize it to minimize a cost function that includes an output error, which is going to be some weighted square of an error term, a difference between what you want and what you have, and an energy term, which is really about here the magnitude of the control input. And now the thing that's remarkable is that under these specific settings, we actually know what the input use should be. And it really should just be some feedback matrix K times the state or activity. Now, for any matrix, you can write it as a product of two other matrices, as long as these matrices have the same dimension as the recurrent connectivity. So maybe that looks a little bit technical, but we can just make it more concrete now, which is that now if we set the matrix A being the cortical connectivity, if we say we don't have any constraints on the direction through which we send the input back, so B is the identity, and we set U and V to be the thalamocortical and corticothalamic synapses, which then implies that the number of units in thalamus basically scales as the number of units in cortex. So you do need a lot of units in thalamus in order to be using this framework at least exactly. Then you can implement this optimal feedback controller with thalamic loops. So this is a very powerful classic mathematical framework with a lot of very mathematically beautiful and very well-defined results. But it also comes with its own set of constraints, which are different from the set of constraints that we consider on the left, notably with respect to basically how many of these units you assume you have access to. Okay, so now I'm going to talk in a little more detail about using these frameworks to model cortical preparation, trigger, and execution, notably during sequences. But before that, I wanted to talk about the experimental background that inspired these models to start with. The first experimental background is that before movement in cortex, several studies have reported that there's a movement-specific cortical preparatory pattern, as well as some movement-independent, at least direction-independent, trigger-like signals that correspond to movement timing. And these appear to be conserved across movement contexts. And there are older papers about that, but for simplicity of the presentation, I'm going to cheat a little bit and talk about more of a newer papers that we didn't quite know about when the models were developed. And so here the different contexts are going to be to have two reaches, one, then two, with a pause in between. You can see the activity of the deltoid muscle here. So it increases during the first reach and then again during the second reach with a pretty steady pause in between. And you compare that to a context where you have a continuous sequence where you really reach continuously, at least at the level of the muscle activity. It just seems to be all mixed together. And then the authors of this study, Andrew Zimnick and Matt Stuchland, did a very clever analysis by really looking at both these preparatory dimensions, which predict movement direction before the movement happens, and which are easily to find in this setup, and trigger dimensions that are about, again, movement timing, but do not reflect movement type. And so what they found is that both in the case of two reaches with pause in between and in the case of a continuous sequence, when they looked at these dimensions, they found that in both of these cases, you were seeing a preparation not only of the first reach, but also of the second reach. In addition, you are seeing two different trigger signals. So the idea is that even though the second reach is now in this continuous sequence, performed in a very different context with much less time, perhaps, to separately think about it, with a very different internal state before you actually performed it. The neural signatures look very similar. And what was very, very striking too is that if you look at the second bump during the continuous sequence, so this pattern here, and if you look at the same bump during the two reaches with a pause in between, you see that the activity very much lines up when you align the reaches. So what that suggests is that when you really have decided to do a specific movement, and it's important here that the movement characteristics look very similar in between the first and the second reach, it looks like at the level of motor cortex, you trigger two separate movements and you reuse the same preparatory activity pattern. And the other piece that was important for the models was the finding that if you think of different movement variants, and most notably most of the reaches that people had been studying so far, like different reach directions, the activity is actually very well captured by a single linear dynamical system here. But the only thing that varies across these different reaches is going to be the initial condition. So what is the initial activity pattern? And the idea is that this initial activity pattern might be what is being set up in the preparatory activity that's shown on the left. So that was kind of like the data and the thinking that people were having more or less at the time where these models were being developed. And now we're going to talk about the actual models, where again, I want to kind of like think about these two different frameworks. So the low rank framework and the more control theory LQR framework. And they have some commonalities, which is that during preparation and trigger, associated with preparation and trigger, in both of these frameworks, you're going to have black thalamocortical loops, as well as an external input X that are going to be optimized such that you converge as fast as possible to a good activity pattern for execution. So in order to schematize that, what I'm showing you in these little plots is really in activity space. So each of these dimensions, you can think of as the activity of one neurons. Of course, it's way higher dimensional than that, but we represent what we can. And these are called flow fields. What they are is that the arrows tell you where your activity is going to go next. And so what's happening here is that the black loops, what they do is that they create all the arrows to go towards a particular point. And the input X tells you exactly where this point is. So with that type of construction, in these networks, you're going to be able to converge fast towards a desired activity pattern for execution. Think about your preparatory pattern. In both of these frameworks here, and that's a little bit different from some later models, but in both of these frameworks, there is such a notion of triggering of movement. Specifically, movement are being triggered by uncoupling black loops and quote unquote disconnecting the input X. None of these models are like very clear on like what the actual mechanism for that is. There could be many different mechanisms, but if you achieve effective uncoupling of the black loops and at the same time disconnecting of X, you're going to trigger the movement. And how accurate it's going to be depends on how close you were to your desired good state to start the movement. So to the convergence point. And one thing that I do want to mention that I wish I could have made a little bit clearer in the article after thinking with more people is that at least the way I think about it is that the input X here, which tells you about which movement you're going to do next, may be produced by premotor regions. And so the idea really, if you think about it, is that this is more of a primary execution circuit that receives this premotor instructive input at the beginning of the movement. The process of this premotor population that prepares this input X could be occurring while this execution circuit does something else, for instance, a previous movement. Okay, so these are the commonalities between these two frameworks, but we've looked before, they have some differences. So here are the main differences. In the Laureline framework on the left, we were really interested in the possibility that for varied types of movements, not just straight reaches, you might need to modulate the execution dynamics. It might not always be the same. It might not be sufficient to just change the initial conditions. And so we thought of how this could be done relatively efficiently by using different thalamic units during its execution. And so as you can see here, it changes your flow field. And so you have more freedom in the way you can generate motor output. It's not just about changing the initial activity in the fixed flow field, it's also about changing the flow field. And at the time, it wasn't quite clear whether that existed. It was just clear that perhaps, like in the model, it was clear that this would help you produce more diverse outputs. On the right, there was more of a focus, and I'm missing an arrow here, but think of here being an arrow. There was more of a focus on thinking of a specialized mechanism to specific later execution dynamics. And because you have many more loops and you know what the future executions dynamics are going to be, then you can account for the fact that there are different directions in activity space that have different motor potencies. So what that means is that like, if the activity is set up right on, say, the yellow dimension, then it helps you a lot to produce a correct output. If you set it along the black dimension, it doesn't actually help you much. And so what these many more loops allow you to do on the right in the LQR framework is that for fixed execution dynamics with this framework, you can prioritize the most important directions for future movement accuracy. So that means that if you want to do as well with the framework on the left, you'll need either a larger input, that would mean larger corticothalamic and thalamocortical weights, synaptic weights, or more optimized baseline or execution dynamics, or a longer transition time. But all of these depends a little bit on the details of the implementation, so it's not quite clear like what's the right, what's the solution that in general might be favored by the brain. And then for the LQR framework, one thing to note is that you could also expand it and kind of like mix it with the ideas of the learning framework and say, okay, now what happens if I do want to learn new execution dynamics? In that case, you're going to have to learn a whole new set of loops here that help you converge towards the right activity state for these specific dynamics, or you're going to have to override the previous one. So in terms of thinking of learning many different types of movements, this comes at a big cost in terms of learned parameters. All right, so I added a little bit of an outro to this to talk a little bit about how to test models. And many different people have many different intuitions about that. And I'm just wanting to share a few of mine. And then very briefly, I'm going to flash some preliminary results from data analysis that I've done. So one way of trying to test model could be to say, you know, like make a model that's like precise enough such that you can do a quantitative fit to a data set and ask which model fits quantitatively better. So this is potentially very powerful. It's definitely very appealing. It comes with some challenges. We all have some challenges, but the challenges, I think, of the quantitative fit is that there is now a well-recognized difficulty of which metrics to use. The quantitative fit also for motor control is going to be affected by which specific movement you instruct the model to do. But, you know, the two models that I showed you, they're more mechanistic models, and they're not really designed to be specific to one movement statistics. They're more thinking about how you can reshape cortical activity for performing accurately many different types of movement. And that's where I think that it's also interesting to think about the fact that there might be a little bit of a difference between vision, which vision neuroscience has pioneered this idea, this metrics approach, and motor control. In vision, the assumption that people are interested in is that cortical activity may extract useful statistics that are conserved across species, as well as the data set, the natural images, is conserved in all the members of the species. And the needs of what you need to do with this data set, we think is the same across all individuals. Say for vision, we want to take natural images and we want to be able to discriminate between objects. But for motor control, the movements are more idiosyncratic to an individual. I'm very short, I'm 5 feet tall. I'm sure my motor control looks very different from other people's motor control. Also, it's not just about the body we're born with. We might have different needs. God forbid, at times we lose a limb, and we learn to use prostheses. We learn to control arbitrary objects that we were not evolved to control, like the pad on an Apple computer. So the movement really is very plastic, and it stays very plastic across the lifespan. And so I think that it's interesting to think about whether there are less constraints from movement statistics than from image statistics if we're trying to do this quantitative fit. And so also, I think that for me, thinking about the way that this came to be, really in the example of vision, the idea that there is an objective for the vision networks, whether you agree or not, but like the hypothesis is like the objective is a fixed task for a fixed data set, whereas for motor control, it's more about maybe we evolve these motor control regions such that they are good for learning. So it's more of a meta-learning process. And so finally, without strong task constraints, what can happen if you start looking at quantitative differences is that they can be affected by many different factors, say the single neuron models, the biophysics, the assumed inputs. And getting one factor correct may only lead to quantitative improvement if all other related factors that interact with this one also are correct. So I think it's not super easy unless we assume that we got enough of it right to really interpret quantitative results. But there's a lot of very interesting work with this. So I think it's great that, people explore this. And for me, I'm also interested in possible alternative and complementary approaches where we are trying to look for qualitative matches between model and data based on mechanistic assumptions of the models that we develop. So what does that mean concretely? I'm going to just briefly mention examples in the literature since I published the models that I feel are qualitatively consistent with the model, and then I'm going to talk about things that I've started to do myself. So the first axis is that it's now much clearer that thalamus is involved in the control of fine movements, that there are large thalamocortical coupling right before the movement, which is similar to the fact that I need a larger thalamic population of neurons to set the right initial activity in my network. It's also true in the other approach. And there is also some beginning of evidence that these corticothalamic pathways are involved in learning and executing movements. Second, and I kind of like presented you the other way around, these models basically assume that you're going to separately prepare each movement. And that's what has been seen in this paper by Matt Schuchland and Andrew Zimnick that I told you about. And finally, people are also starting to see that even though for straight reaches, a single linear dynamical system was doing well to fit neural activity in these different reach conditions with different initial conditions. When you consider more complex movements, such as curved reaches, then actually you don't do as well if you assume just a single linear dynamics. And then you're going to see that there are actually changes in these dynamics. These can be characterized as changes in the basis function characteristics, but the position of the basis functions, they found very strong changes there. And also, more specifically for some of these basis functions, they showed some changes in the frequency, more in the supplementary material. So this is some evidence that it's perhaps important to think about different execution dynamics. But I think that there is more testing that can be done. This is the last slide. So I'm very lucky that I was given a chance to look at data that was collected by Stefan Lenke, who's been very helpful to discuss the data, and Adam Huntman. And it's projects that I've been able to work on with students and interns that come to MIT, Alex and Ria, and under the guidance of Hila. So the idea is that I have access to recordings from motor cortex and thalamus, and this is while mice are going to reach towards pellets. It's a cortical dependent task. It takes many, many days to learn, 5 days to learn for the mice. And the questions really that we want to ask is, in this reach, grasp, retract, and eat sequence, are there hidden behavioral motifs that compose the reach? Can we find neural signatures of separately triggering and preparing each motif? Do switches in thalamic activity precede switches in cortical dynamics in the way that we saw in the models? Are the thalamic populations that are involved just before a movement shared across motifs? That was a point of difference between the low-rank framework and the more control theory framework. And does learning without interference involve new move-specific thalamic units? So I don't know how many of these we can tackle, but at least I started tackling the first and the second point. And what I did is I look at a principal component analysis that maximizes behavioral variance across trials. So then for each trial, we can project back on a specific principal component axis, which gives us a PC score. And then each trial, we can plot back in the original behavioral variables. Here it's a X and Y position of a finger. And this is the analysis window that I used to try to find coherent correlation patterns across trials. And as you can see here, the first PC captures mostly the pre-reach variance. So the mice, they start from a little bit of a lever position, and there is variability in the position in which they start. And then interestingly, for the second PC, what happens is that now the trials are going to be very much ordered into the PC score during the initial reach. And they're going to be ordered until a certain point that appears to be slightly beyond the analysis window that I used. So could that be submotive? So this is not, this is just behavior. That's consistent with a lot of many different neural hypotheses. One of the things that I've started to look at is the cortical trigger dimension. So this is going to be a dimension that is trained to make the difference, to differentiate between pre-reach and post-reach. The training data that we used for this dimension is here. It's really right before you start reaching, and then right after you start reaching, more or less at the same time here as the PCA data that I used. And so the interesting thing is that as you expect, it increases on average across trial at the beginning of the reach. We trained it for that, then it decreases a little bit, and then it re-increases a little bit. So the question is that, do we have a second peak here, as there was a second peak in the Churchland data? And to start getting some evidence for that, we can median split the trials between those that are fast and slow. And here, fast and slow, the thing that's good to and easy to quantify is the start of hand opening that starts while the mouse is going towards the pellet. And so what's happening here is that, as you would expect, if this is indeed a second trigger, for the slower trials, the second peak is going to be shifted, but it's not shifted at the beginning. So this is preliminary. We've seen that in 2 mice and I think 8 sessions. But this is really an important start. If this is confirmed, then I can move ahead now that I have different motifs. I can look at whether there are separate thalamic populations that are involved in these different motifs and look at the associated cortical dynamics. So I want to thank you for your attention. Thank you very much for inviting me and giving me this opportunity to discuss these topics that I'm very passionate about with you. And I want to thank Sean and Kaushik, who wrote the review with me, my experimental collaborators, Stefan and Adam, the interns that helped me with the data analysis, Alex, Ria, as well as Ela, for giving me feedback. I've received feedback notably for Sartak and Chris and a lot of different people along the way. And I want to thank the Salmon Foundation for the funding.
Thank you.
All right, thanks for the great talk, Laureline. Lots of food for thought. Any questions?
Yeah, it was.
I have a, hi, Laureline. Good to see you. I have a really coarse question. So your models all depend on that thalamus is kind of like a modulator on cortical dynamics. Now, if you deactivate all thalamic cells, doesn't the cortical dynamics completely break down, at least transitory? I don't know what that lesion, like, I thought I learned that, you know, if you silence the thalamus, everything goes to sleep in your cortex. It's a very powerful, it's not just like a modulator on cortical dynamics. There is no cortical dynamics without thalamic input. Is that true, or what's the newest data on this?
I think it depends a little bit on the task. That said, like even in the models that I suggest, what's happening really is that without the cortical units, and I didn't get into detail, what I propose is that everything does go very quickly to zero. And it's only when you put the thalamic units that then you push some of these basis functions to have longer time scale, and you see things that are more sustained. So some things that you can do here. It's a modulator in the sense that like you don't push all of the basis functions to be very slow. You push a low dimensional set of them to be very slow.
But from the experimental side, yeah, so that makes sense. But from the experimental side, that what I learned is still true, that if you shut down the thalamus, the cortex very quickly shuts down as well. Is that, I don't know what the newspapers are.
That is true for premotor cortex. So there is data from Karel Svoboda on that. In motor cortex, I don't know that the effect was as drastic. It definitely has a strong effect. It can prevent movement start. It can affect movement execution, but at least the paper was not written with as drastic of a tone, if that makes sense. It does have strong functional effect, but I don't know that like I don't remember the message being, oh, it's now all immediately zero. Does that make sense?
Yeah, okay, great, thanks.
Other question?
Yeah, I can ask one. Thanks for the nice review of the different types of the models you had in the beginning. But while you was like explaining them, I was just thinking constantly that for the type 1 and type 2 that you presented, they kind of have predictions that are completely rejected in our data these days, at least. So for example, the type 2 model predicts that you should have units that are specifically active in one context, but completely inhibited in others, right? So does that mean that we should just immediately drop these types of models and like go forward with the other ones? How do you think on that?
You know, I focused on the type 3. I think it's just a little bit tricky at times, like, you know, at times some models, like they're not very evidenced, but you can get an insight that then gets useful for something else later. So, you know, I wouldn't necessarily shut down people to think about, you know, different ideas. But I agree with your I personally agree with your assessment that it seems to me so far, like I feel pretty convinced that it's important to think more in terms of thalamocortical loops, and it's important to think more about motor cortex being reused, being this set of parameters that can be reused across many different motifs. So,
thank you.
Yeah.
I have a question. In these, when you're focusing on the thalamocortical loops, how does the sensory input that gets driven through thalamus, either from other structures or straight from the periphery, play in? I mean, you know, like in your experimental example, you're talking about a mouse that's reaching to a pellet, and you have thalamocortical loops transition between substates. That makes a lot of sense, but so does, for example, extremely potent peripheral input that will drive through thalamus at the moment of contact with the pellet, for example.
Right. So I think that there is an interesting point to be made about very large effective changes in cortical dynamics across large chunks of movements, right? So like, for instance, you have this conserved preparation and trigger in the two separate reaches versus the reach sequence. The sensory inputs are going to be very different there that happen at the moment of triggering the second reach. And yet you have some conserved type of events in motor cortex. And so that assumes that, like, at least on top of effect of just sensory feedback, there is something that's more about motor primitives, motor motifs, something about how we break our behavior into reusable segments. So that's what these models focus on. I think it's there, but that means that also, that doesn't mean that there are not other things. I definitely believe that there are other things, and also motor cortex is not alone in, generating the motor output. So does that make sense, what I tried to say?
Well, yeah, it totally makes sense, but I'm just wondering if the mechanism by which the one operates, sometimes you can have these subdivisions that are based on very clear, potent, or very, very time-defined sensory inputs, and sometimes you may not, right? And often they could happen, they could be both in play. And I'm just wondering if you envision them to be kind of highly overlapping and how they work in that scenario, or are those two separate paths through the thalamus?
I would expect, intuitively, I would expect some separation, because I would expect those two different types of inputs to work on very different timescales and to require very different types of processing. So the more like sequencing input is going to be more about planning, about premotor activity. It can go through a lot of different places. So, premotor activity could come back to cortex through thalamus. It could come back to cortex through thalamus, through cerebellum, through basal ganglia. To be honest, I don't know that we have super strong evidence of exactly where it goes through, but it's probably separate from whatever goes on the fast sensory feedback scale. It's be my suspicion, if that makes sense.
Interesting. Okay, we're out of time. Is there one more question if we have it? Otherwise, we'll wrap up. Okay, thanks again, Laureline, for joining us. That was wonderful.
Thank you so much for the invitation. Thank you, and the discussion.
Thank you very much.
Thank you very much. Bye-bye.
Bye.